So good morning, my name's Glenn Thorlby and I'm a research scientist at Scion working in projects involving tree biotechnology. So as you can see from the title, today I'm going to talk a little bit about gene editing and its potential uses in forest productivity, resilience and sustainability. And to start with, I want to really put this into a little bit of context about why we're doing this work. And central to this is the information that you can see in this slide. And this is really that mutations, or the rare errors that occur during DNA replication, are what leads to genetic variability. And so ancestors which happened in either you or your ancestors define what you are. And these mutations are really the basis of natural selection. And in this slide you can see a collection of birds with different shaped and coloured beaks. And these beaks have occurred by natural selection, and it's actually been a whole series of different mutations that have accumulated over a long time period to give this degree of variance. But this can happen over a shorter term period, and you can see that by looking at the moths on the left of the screen, and if you can make out one of these moths is black and the other's white. And the black moth developed with a single mutation which occurred during the Industrial Revolution in Britain when pollution was making everything very black and camouflage was better if you were black. And of course these mutations are also the basis for artificial selection or breeding. And rather like the birds, some of these have taken a long time and involved the build-up of a lot of mutations. This is the case with corn, which you can see in the little picture here, which was first domesticated about 10,000 years ago. And the corn we see today doesn't really bear any resemblance to its ancestor. And that's because a whole series of different mutations have been selected over a very long time period. But there are also examples of domestication where the changes haven't been so profound and so many mutations haven't been needed. And brassicas that we eat are a good example of this. And all of the examples you can see in this other part of the slide are actually the same species, and they have relatively few mutational differences between them. But these give different structures which have led to different crops developing. By about the 1950s, it was become apparent in a lot of crop breeding programs that there wasn't actually enough variability available to develop the crops further, which was what was needed in order to get higher yields to feed the growing population. So methods were needed to generate more variation. And a method that you can use to do this is called mutagenesis, and this leads to mutation breeding. A mutagenesis is simply a technology which allows you to increase the rate of mutagenesis. So going from that low background level to higher levels so that you can actually have more variation. And there are several ways you can do this. You can do it with either chemical or physical mutagens, and a lot of different methods have been tried. On this slide I've put an example of one of the more interesting ones, which is radiation breeding. And this is what's called a gamma garden, and these used to be very common, but now there are very few left. This one still works in Japan, and at the centre of this garden is a radiation source which emits gamma rays. And in order to carry out mutagenesis, plants are planted in this garden. Near to the source they get a lot of radiation, further away they get less radiation. And after a period of growth, these are taken away and then examined to see if they've got any interesting phenotypes. But the problem with mutagenesis breeding is that it's a random process. Mutations are generated at random right across the genome. And so you have to mutagenize a very large population and screen it to find interesting mutations. And even if you find one, the chances are that this mutation will be accompanied by undesirable mutations in the genome. So you have to do a lot of crosses in order to sort out what you want from what you don't want. This makes this fairly unuseful in conifers, shall we say, because of the long life cycle, we don't have the chance to do all of these crosses to purify things, so it's not practical to use it. However, it has been very useful, and over 3,000 mutant varieties have been developed, and many of these are actually in the foods we eat every day. And I guess this is where gene editing fits in, because gene editing is really a way of creating designer variation. We're actually changing a gene that's already in the genome, so it's a form of mutation. And in fact, the mutations you get are of exactly the same type if you get natural mutations or if you use these mutagenesis protocols, and so you can't distinguish them. This has led to many parts of the world regulating this sort of gene editing as a non-GMO, although in New Zealand it is currently regulated as a GMO. But the real advantage of this technology is that it allows you to make highly precise targeted changes. You don't have this random process. You can actually go into the genome and change a particular gene. So it's knowledge-based, and you can go in, change the gene you want, and hopefully then get the phenotype you want. And this slide just shows you how this process actually works. And I'm talking here about CRISPR, which is the gene editing technology that we use at Scion, but there are a number of gene technologies you can use. This is a very fast-moving field, and new ones are developing all the time. But in the case of CRISPR, the mutagen, if you want to call it that, is actually an engineered nuclease, something that we develop. And it comprises of two different elements. In orange, the DNA targeting part, and then another part, the nuclease, which is really something that cuts DNA. And you design the targeting part to a particular sequence of DNA, and this directs the nuclease to exactly that place in the genome. And when it arrives, the nuclease cuts the genome at the precise point you want, and that's the first stage of making a mutation. And so next I wanted to just mention some of the advantages of using gene editing in forest trees. And by forest trees, I'm particularly talking about conifers grown in New Zealand's planted forests. And I guess the biggest advantage is that forest trees are undomesticated. They've had very few rounds of breeding. As you can see, 3 to 4 in most cases. This compares to the thousands of rounds in things like corn. And because they've only had these few rounds of breeding, there hasn't been an opportunity to bring together all these desirable mutations to get phenotypes we would like. In fact, we don't even have the collection of mutations we would like. And so gene editing allows us to overcome this in a very speedy and rapid way. Because if we have a gene where we know that we would get a good phenotype by changing that gene, we can actually go and do it in one round. We don't change the rest of the tree, and so we can very rapidly introduce new desirable changes. And there are many applications that we can use gene editing for. And if you follow the popular press, there's these days often a story about a new gene-edited crop which is about to be tested or released. And many of the same things can be done in forestry. And key traits that we could improve are things like forest health, where we can gene edit to give greater disease resistance. We can also improve productivity and sustainability. And for the majority of the remainder of the talk, I'm going to talk a little about a sustainability trait, particularly dealing with the wilding problem in New Zealand. And that's because we've just finished a large program looking at this, and it was partially funded by the forest growers. But before I get to that, I wanted to say a little bit about the technology we've actually developed at Scion and how we're using it. And as you can see on this slide, we've actually developed editing and edited several genes in both Pinus radiata and in Douglas fir. And as I'm going to say in a moment, we've edited Douglas fir for sterility genes as part of the Winning Against Wildings project. But we've also done a lot of editing in Pinus radiata, and as it says on this slide, we've edited several genes which are related to the structure of cell walls, and we've also edited some genes related to sterility. And you can see a picture here of some Pinus radiata trees which actually have edits in a cell wall gene, which we're growing on a glasshouse to see if the phenotype's what we expect. But for the range of talk, I just want to say a bit about the Winning Against Wildings project. And as I said, this has been a 5-year programme which has recently finished, which was partially funded by the forest growers. And this project really encompassed all aspects of the wilding problem in New Zealand, and there was a focus both on the existing wilding problem and on preventing future wildings. And it was in this prevention aim that we worked. And our focus was on Douglas fir, and the reasons for this are that Douglas fir is a commercially important species. It's planted commercially, unlike many of the trees which create wildlings, and it's also very wilding prone. And so if we're to continue to be able to plant Douglas fir, we need strategies to make sure it doesn't generate future wildings. And we took 2 basic approaches to do this, which I'm going to talk about today. The first of these was the propagation of naturally low coning lines. And in Onslow 1 seed orchard, they had some trees which they'd identified as being low coning. Year on year, they produced less cones than the other Douglas fir trees in the orchard. And we wanted to propagate these trees, see if they preserved their low coning nature, and test them out in the environment. And the second thing we wanted to do was use the gene editing technology I've just spoken about to see if we could use this to develop some sterile trees which couldn't generate wildings. And so at the start of the project, we had to develop some technology to enable us to do it. And the first technology we had to develop was some tissue culture capability, because at the start of the project, we didn't have a plant regeneration system for Douglas fir. And we successfully did that in the first couple of years. And you can see along the bottom of this slide what we developed, which is a cell culture system, allowing us to go from some cell cultures through somatic embryogenesis through to some trees, which we can have out in the glasshouse and ultimately the field. We also needed to identify a set of genes that are essential for reproductive development. And that's because our strategy was to gene edit these genes so that they were no longer working. And if you took out an essential gene, they wouldn't be able to reproduce, and you would have sterile trees that couldn't generate wildings. And before I summarise what we've done with gene editing, I just want to say that the low coning strategy has been successful. We've managed to propagate these trees. They're about to go to Scion's nursery for some further growth before they're field tested by Onslow 1 in some different environments to make sure that in each environment they're still low coning. A challenge with this is it's a long experiment. We have to wait for them to reproduce. And really my final slide is just to tell you where we've got with the gene editing. And as you can see here, we've successfully edited 3 different genes we believe are essential for reproduction. And the first of these genes, or the first one which we edited, DAL21, now has trees ready to go into the glasshouse. And you can see some little pictures of these trees on this slide. And we're going to plant them in the glasshouse so we can check that they grow normally. Because although we think we've edited reproduction, we want to make sure we haven't affected anything else and that the trees grow normally apart from being affected in reproduction. We have these lines stored in cryopreservation so that we're ready to use them in future if we need to take them out again. The big test that we have in this project is how we're going to test sterility. Because, like the low coning trees, it'll take a long time, and currently in New Zealand there isn't a regulatory route to allow us to do this. We can't grow these trees outside because they're classed as GMOs. So we can't grow them to the stage where they reproduce and test whether the reproduction works. So we're a bit of an end here. We have to really wait for the government to change its legislative stance or look at doing this overseas. And to finish, I would just like to acknowledge the people who have actually done this work rather than me talking about it. And these are listed on the slide. Charleston and Laurel have done a lot of the CRISPR design and development. Steffi has done a lot of the screening, and Barbara and Kathy have been instrumental in developing the tissue culture technologies.